Ondansetron, the antiemetic drug. So what does it mean to be an antiemetic drug? It simply means to prevent nausea and vomiting. In this case, ondansetron is a specific example of a drug used in preventing vomiting brought on by the distressing effects of chemotherapy. So why do we need ondansetron? Well, firstly, chemotherapy is administered intravenously directly into the bloodstream, where the chemical agents within the chemotherapy travel up to the medulla oblongata, which is found at the base of the skull. The chemoreceptor trigger zone is found here and receives the input from drugs such as chemotherapy, causing a reaction, in this case, vomiting. Specific chemotherapy treatments causing nausea and vomiting include cisplatin and cyclophosphamide, which both cause severe nausea and vomiting. The vomiting centre is also found in the brain, and there are a number of signals that can result in vomiting specifically. Firstly, signals from the chemoreceptor trigger zone, known as the CTZ, react to chemicals or drugs in the blood, such as chemotherapy. Secondly, signals from the brain cortex and limbic system can react to sight, taste, and smell, or emotions and pain. Thirdly, signals from other organs and nerves can respond to disease or irritation in these organs. In chemotherapy, areas in the esophagus, stomach, and intestines could be triggered. Some of the risk factors associated with nausea and vomiting when undergoing chemotherapy include if vomiting has been experienced in past chemotherapy treatments, if you experience motion sickness, vomiting with pregnancy, you are a younger patient or a female patient, if your fluid balance is abnormal due to dehydration or bloating, if you experience constipation, especially when taking opioid drugs, if you suffer from recurring infections, if you have kidney diseases, or depending on the location of tumours, for example if they are found in the digestive tract. Nausea and vomiting can occur at any time during chemotherapy. It can occur within 24 hours of starting chemotherapy or later. If it occurs in the first 24 hours, it is known as acute, and if it occurs later, it is known as delayed. The sight and smell of chemotherapy can lead to something known as anticipatory nausea and vomiting, which occurs before chemotherapy is delivered in those who have experienced vomiting in prior cycles. Ondansetron was invented in the early 1980s by a leading scientist called Sir David Jack. Jack's career as a pharmacist and drug discoverer began as he started an apprenticeship in Boots the chemist, before undertaking an honours degree in pharmacy and pharmacology at Glasgow University. He gained a PhD from the University of London. In 1961, he joined GlaxoSmith as head of research, where he remained until his retirement until 1987. Ondansetron is known as a 5-HT3 receptor antagonist, meaning it blocks the action of the 5-HT3 receptor. The discovery of ondansetron stemmed from Jack's focus on the central nervous system and the gastrointestinal tract. The drug was first launched under the brand name Zofran in 1991. Shown here is the chemical structure of ondansetron. The following functional groups are present: an imidazole ring, a cyclic ketone, and a 5-membered nitrogen-containing ring. So does the drug fit Lipinski's rules? Well, firstly, the molecular weight is 293.37. Secondly, there are no hydrogen bond donors, there are 2 hydrogen bond acceptors, and the log P is 3.9. So overall, the molecule does fit Lipinski's rules. As we have already said, ondansetron forms part of the 5-HT3 receptor antagonist family. 5-HT3 receptors are an example of ligand-gated ion channel receptors, which are part of the cis-loop family. Here is the specific ion channel receptor, composed of 4 transmembrane spanning helices. The helices are composed of 5 subunits surrounding a cation pore filled with water-permeable ions, these ions being sodium, calcium, and potassium. Individual subunits each hold an amino acid chain labelled M1 to M4, which are connected by extracellular and intracellular loops. Serotonin is the only class of family with receptors identified as ion channels. The N-terminus can be identified by the NH3+ group, and it contains a tryptophan residue, which is essential for ligand binding. The C-terminus is identified by the carboxylate anion. Shown here is the pharmacophore for the 5-HT3 receptor antagonist. The A ring is shown at an optimal distance from the oxygen. However, the A ring only acts as a spacer and is not actually required for binding. Ring B, however, is much more important and links to hydrophobic binding regions. As you can see, the ondansetron structure overlays the pharmacophore, which displays their structural similarities. So now we need to consider how the drug ondansetron works on the 5-HT3 receptor specifically and its mechanism of action. The serotonin 5-HT3 receptors are located on the nerve terminals of the vagus in the peripheral nervous system. The action of the chemotherapy agent stimulates the release of the serotonin molecules. The serotonin stimulates the vagal nerve receptors to trigger the vomiting center in the medulla oblongata and the 5-HT3 receptors in the chemoreceptor trigger zone. This initiates nausea and vomiting. Ondansetron works to competitively block the action of the serotonin molecules at the receptors, and this results in the inhibition of the chemotherapy-induced nausea and vomiting. The first 5-HT3 receptor antagonist discovered was ondansetron. This was modified from cocaine. The cocaine molecule itself showed minor antagonistic behaviour on the 5-HT3 receptor. The discovery of ondansetron allowed hundreds of other drugs to be formed from it, which eventually led to the discovery of ondansetron. Now we are going to discuss the three syntheses for ondansetron. Synthesis 1. A condensation reaction occurs between the 2-bromoaniline derivative and the diketone molecule, which form molecule 2, called an aromatic amine. This is carried out under heat, for example at 120 degrees Celsius. Methyl iodide is introduced as a reagent to form the methyl group on the nitrogen, and bromine is then the leaving group. An intramolecular cross-coupling reaction occurs in order to form the indole functional group, which has an additional cyclic ketone attached. Several steps are then indicated by the overlapping arrows because ondansetron wasn't created in a single step. Synthesis 2. There are two different methods which can be used to form the imidazole ring shown, which contrasts to the first synthesis in which the imidazole ring was created last. It can use either basic or acidic conditions, and for both of these the yields is particularly high, being from 90 to 95%. Alkylation of the imidazole ring with molecule 10 then goes on to form ondansetron. Synthesis 3. A strong base such as butyl lithium is used in the first reaction to form a quaternary amine. Methyl iodide then acts as a methylating group to form a methyl group on the nitrogen ring, and then compound 15 will tautomerize. Then a condensation reaction occurs, and this is seen by the loss of water. This is due to a reaction between a ketone group in compound 15 and a hydrazine, which can be identified in compound 16. The last step is the Fischer-indole synthesis, and this forms ondansetron. The second synthesis has a yield of 86%, in comparison to the third synthesis, which only has a yield of 55%. The synthesis with the yield of 86% is therefore much more favourable. On-target and off-target effects. On-target side effects are defined as effects related to the mechanism. In this case, the pharmacological effect on the serotonin receptor-related 5-HT family. Off-target side effects are those which are unexpected. For ondansetron, this includes binding to receptors outside of the 5-HT family with no evidence as to why this happens. Common or very common side effects. These can include constipation, feeling hot, and having headaches. Feeling hot and experiencing headaches is expected as a result of ondansetron acting on receptors in the brain. The receptors associated with these side effects are thermoreceptors in the hypothalamus. Uncommon side effects. These can include arrhythmia, chest pain, and seizures, as well as hiccups. This occurs due to damage in the vagus nerve, and they can cause hiccups to last for a long time. This is because ondansetron acts specifically on the vagus nerves at the 5-HT3 receptor. Rare or very rare side effects. These can include dizziness, vision disorders, and QT interval prolongation. QT interval prolongation is a rare side effect from the clinical use of ondansetron, and is said to exert its effect on the cardiac cycle, as it is able to interact specifically with the inactive sodium channels and cause some of them to become blocked, in turn prolonging the QT interval and delaying the flow of potassium ions out of the heart, meaning too many sodium ions remain inside the heart. In summary, why do we need ondansetron? Up until 20 years ago, nausea and vomiting were side effects of cancer treatment that were unavoidable. Now this side effect can be prevented in up to 80% of patients with minimal side effects caused from the use of ondansetron itself.